Today's lecture is dealing specifically with the complement system. Up until this point in the semester, we've discussed quite a bit of detail about the innate immune response and the adaptive immune response, and throughout the lectures that we've had to date, I've mentioned the complement system quite a few times, and I've also mentioned that we will eventually get to talking about this third facet of the immune response. What we discussed with respect to the innate immune response and the adaptive immune response is that both of those mechanisms can actually activate or initiate the complement system. When we discussed both innate and adaptive immunity, we discussed specific cell types that dominate in one or both of those different areas. What we see with the complement system is that many of the cells that we talked about play a role in this particular response. In addition, I also want to mention that the complement system got its name because it was thought to help or complement the clearance of pathogens in both the innate and adaptive immune response. However, we know again that this particular third facet of the immune response is an important response altogether. As we'll see in just a bit, the complement system is actually a very complex biochemical cascade. But what we can do is assign functions associated with that complex biochemical cascade. Those functions are listed here for you. The first being lysis, that is cell lysis. What we see is that there is a complex that is formed in response to initiating the complement system known as the membrane attack complex or MAC. In general, this mechanism is a mechanism of perforating the cell membrane of a given pathogen and eventually leading to the destruction of that pathogen. Another function of the complement system is opsonization. We've talked about opsonization as well in the first half of the semester, and opsonization is a mechanism of tagging or targeting a pathogen for the eventual destruction via phagocytosis. A third function of the complement system is to activate the inflammatory response. Again, we've seen the benefits of initiating an inflammatory response and how that can fight off a given infection, while the complement system also plays a role in initiating additional inflammatory responses. Fourth, we see that the fourth function of the complement system is the clearance of immune complexes. Now, this is something that we didn't discuss in too much detail when we considered the adaptive immune response, but we know that antibodies do in fact associate with pathogens and form complexes with these pathogens. It's one of the functions of antibody responses. Well, with the complement system can recognize these immune complexes that have formed with antibodies and also initiate or trigger phagocytosis. And then lastly, what we see as a function of the complement system is viral neutralization. We discussed how antibodies, again, can act as neutralizing agents, while we know that components of the complement system can have that similar type of function of neutralization of viral particles. So what I'd like to do first is talk a little bit about the components that we find in the complement system. The components of the complement system are mostly proteins and some glycoproteins. There are over 25 different protein and protein fragments that have been identified as part of the complement system. These proteins and glycoproteins are synthesized mainly by the liver. However, there are some examples of immune cells, leukocytes, that play a role in synthesizing some of the complement system proteins as well. Macrophages, epithelial cells, and monocytes are included in those leukocytes that do have the ability to synthesize complement system components. But again, just to reiterate, the majority of proteins that are part of the complement system are synthesized by the liver. In addition, it turns out that complement system proteins make up about 5% of the proteins found in the serum. Lastly, I want to mention that most of the complement system proteins circulate in the bloodstream of the host system as something referred to as a zymogen. Now, a zymogen is basically an inactive precursor, also sometimes referred to as a proenzyme. What has to happen is these precursors need to be activated in order to function as part of the complement system. The activation of these proteins occurs typically by cleavage of the original protein into subcomponents, and these subcomponents then represent active complement system proteins. One question that is oftentimes asked is, why are these complement system proteins circulating as an inactive form? And the reason for that is that those inactive components are basically ready to go, waiting for the initiation or the activation mechanism to trigger that response to continue. So having inactive precursors allows for a very quick response when triggered by either the innate or the adaptive immune response. Now, because there are so many different components associated with the complement system, I want to go over a little bit of the nomenclature associated with these proteins. Typically, most complement system proteins have the letter C and a number, like you see here, C1 through C9. Some complement system components do have other letter designations or name designations as well. There's a few, something like factor D is an example. Lastly, just to complicate things for us, some of the complement system components also go by their trivial names, for example, homologous restriction factor. Now, again, a majority of the components do have the C and the number designation, like C1, C2, C3, for example. And the cleavage products that are produced in the activation of these inactive precursors lead to the A and B designation. So, for example, C3 is cleaved to form C3A and C3B, and we'll see that in just a minute with a few slides down the road. As I mentioned at the beginning of the lecture, the complement system is a complex biochemical cascade, and we will look at that in just a minute. However, what I typically like to do is break it down into smaller chunks so that we can easily understand what's going on. What we'll start off with on this particular slide is looking at the activation pathways that allow for the activation of the complement system. There are, in fact, 3 distinct ways that can activate the complement system. There's something known as the classical pathway, the mannose-binding lectin pathway, or MB-lectin pathway, and the alternative pathway. The classical pathway is the only one of the 3 that does require antibodies to help with the activation mechanism. The other 2 occur without the help of antibodies. We can expand that figure from the last slide and include a lot more detail in this complex biochemical cascade. The important thing to note is that there are 3 different ways to activate the cascade. In this particular slide, you're seeing, again, a lot more detail with specific names of components that are identified in different pathways. What is important here, though, is to actually focus on functional protein classes that components of the complement system fall into. So rather than learning all of the different components and everything that they do, what I'd like to do is break it down into some broad categories of functional protein classes. So again, if we have all of the complement system components, we can categorize them by what they do or what their job is in the complement system. What you can see here are the 7 broad categories of functional protein classes that individual complement system proteins can fall into. The first one, or category number 1 at the top of the slide, are those proteins that are considered initiators. These are proteins that are involved in activating the complement system. We will look at each one of the pathways in a little more detail, and we'll look at some specific components that fall into this initiator category. But again, any protein that helps with the activation of the process would fall into this functional category. The next one, number 2, are categorized as convertase activators. There is a key enzyme or several key enzymes known as convertases. These are enzymes that help convert the inactive components into the active components. Like I mentioned previously, this typically involves a cleavage of the inactive component to form an A and a B subcomponent. The enzyme responsible for this cleavage is typically referred to as a convertase. So, any complement system component that is acting as an enzyme to cleave one of the inactive components to form active components would fall into this particular functional category. Category 3, opsonins. Well, this is something we've heard lots about previously. Opsonins are molecules that interact with the pathogen, bind to the pathogen, and tag that pathogen for phagocytosis. So any of the complement system components that act as opsonins would fit into this particular category. Number 4, anaflatoxins. Now this is a term that we have not utilized previously. We have discussed what the result of anaflatoxins are. These are components that actually play a role in inflammation. So one of the jobs of the complement system or functions of the complement system is to help with the inflammatory response. So any of the protein components that help with inflammation or initiating inflammation would fit into category 4. Category 5, the membrane attack complex. Again, as I mentioned at the beginning of lecture, there is a specific component of the complement system that is involved in cell lysis, and this occurs by basically putting holes in the membrane of the pathogens, and this is accomplished by something known as the membrane attack complex. So, any of the proteins that are part of the complement system that play a role in forming the membrane attack complex fit into this category. Category 6, complement receptors. Any of the complement system components that act as a receptor to receive information and allow for cells to act in a specific way, just like we've talked about receptor-ligand interactions throughout the entire semester, would fall into this category. And then lastly, category 7 are complement system components that are considered regulatory proteins. This facet or branch of the immune response is actually highly regulated. In particular, we have discussed previously that prolonged periods of inflammation are not necessarily a good thing for the host system. It's a good thing to fight off a pathogen, but having prolonged periods of inflammation can cause cellular damage, tissue damage, and the like. So there are proteins that act to regulate this mechanism so that it can be turned off relatively quickly Just like we talked about turning it on relatively quickly, there are also proteins that play a role in turning it off relatively quickly, and they would fit into this final category. So now that we've looked at the complex mechanism, let's go ahead and distill that information just a bit. So what I want to do is start off with a simplified view of the activation pathways that occur to activate the complement system, and then what we can actually boil it down to as the functional response of this activation. So the first thing I want to focus on is what's known as the classical pathway. So the classical pathway is a pathway that in fact utilizes antibodies plus the C1 macromolecule to recognize the microbial component. And once that recognition with antibody as well as a C1 macromolecule complex, and we'll look at that macromolecule complex in just a minute, but it's both antibodies and complement system components, specifically known as the C1 macromolecule, interact with each other to recognize the microbe and trigger a series of events in a signal transduction cascade. Now the arrow that I have here is just a single arrow. It doesn't mean that it's just one step that occurs from this point to the next point I'm going to show you. There are multiple steps that occur, but for simplicity's sake, I've left it as a single arrow. So what happens once the antibody and the C1 macromolecule interact and recognize a microbe? This leads to C3 and eventually C3 conversion into active C3A and C3B. But we'll get back to that in just a minute. The next activation pathway is referred to as the alternative pathway. Now, with the alternative pathway, there are no specific components like we just talked about with the classical pathway that interact with the microbial pathogen. There is or are recognition components, but it's not a specific component like we just discussed. The microbial pathogen is recognized. A series of steps occurs to the eventual convergence at C3. Lastly, we have the mannose-binding lectin pathway. Here too, we have microbes being recognized by a protein known as mannose-binding lectin. Mannose-binding lectin interacts with the microbe, and this then triggers a signal transduction cascade and a sequence of events also leading to C3. What happens once we get to C3? Actually, let me take a step back. So all three of these mechanisms are activation pathways to get to this point. C3 conversion into C3A and C3B. The key enzyme that does this conversion, again cleaving C3 to produce C3A and C3B, is known as C3 convertase. So what happens once C3A and C3B are produced? C3A, again, just a single arrow here, but multiple steps are involved, will lead to inflammation and phagocyte recruitment. C3B, again, just a single arrow here, but leads to multiple steps that will eventually lead to opsonization. And lastly, again, a single arrow, but C3b also initiates the formation of the membrane attack complex. So as you can see here, again, we have a much more straightforward, streamlined biochemical cascade looking at the activation pathways. What's really important is that conversion of C3 into C3A and C3B, and then the downstream effects of that, which are inflammation, phagocyte recruitment, opsonization, and the formation of the MAC. So, as I mentioned with the last slide, I wanted to take a little bit closer look at the classical pathway and how exactly the activation mechanism takes place. There is what's known as the C1 macromolecule, and as you can see here in this figure, it's actually made up of multiple different proteins interacting to form this stalk-like structure. The C1 macromolecule recognizes and binds to the Fc portion of the antibody, and you have the Fab portion of the antibody binding to the antigen that's part of the pathogen. And so it is this complex antibody, antigen, C1 macromolecule interaction that then triggers this particular pathway of activation. So let's talk about each one of those pathways again, just in a little bit more detail. The classical pathway is triggered by antigen-antibody interactions, or more simply put, immune complexes that form with the association of the C1 macromolecule. Now it turns out that IgM and IgG are the only two antibody classes that trigger this particular activation pathway. And IgM is actually more effective in triggering the activation of this pathway in comparison to IgG. Now, from this particular slide, all I really want you to focus on is that important step of conversion of C3 into C3A and C3B. And it turns out that this conversion will eventually lead to another series of steps that allow for the formation of the MAC. So again, overall, what I want you to know from this particular slide is the importance of C3 conversion into C3A and C3B. The mannose-binding lectin pathway has similar components or similar activation mechanism as the classical pathway. You have a protein known as mannose-binding lectin that interacts with surface components of pathogens. As you see in this particular slide, we've got a polysaccharide antigen that is recognized by the mannose-binding lectin component. Specifically, the polysaccharide is mannose, hence the protein that's binding is mannose-binding lectin. Once you have this interaction take place, this allows for the activation of a serine protease. So this is a specific type of enzyme that has the ability to degrade or cleave or break down proteins. In particular, this activation of the protease, which is known as a MASP, mannose-binding lectin associated serine protease, will allow for cleavage of other complement system components, and those subsequent steps are initiated like we talked about in the previous slide. Now, the alternative pathway is initiated by cell surface components, typically polysaccharides, and again, we're talking about cell surface components on the pathogen. These can be things like bacterial cell wall components, fungal cell wall components. There are actually some viral components that can be detected by this alternative pathway, parasitic components, and tumor cell components. So again, you can see that this is an all-inclusive type of activation process. There are 3 different subcategories in the alternative pathway. The first one is known as the alternative tickover pathway, and this is a pathway where small amounts of C3 are always cleaved but inactivated, and if there's no infection, then there's no need for this particular component. But in this case, there's always a small amount sort of just ready to go, ready to fight off a pathogen. You also have the alternative properidin activated pathway, and properidin is a molecule that actually stabilizes the components of the complement system. And in this subcategory of the alternative pathway, the properidin activated component can directly bind to these cell wall components or cell surface components of the pathogens. Lastly, we have the alternative protease activated component or pathway, and this is actually a clotting cascade that's activated by inflammation, typically damage and trauma. So again, 3 subcategories for the activation of the alternative pathway. So we've discussed the fact that C3 gets converted into C3A and C3B. C3A leads to inflammation and phagocyte recruitment. These are all things we've talked about in the past, so this is nothing new for us as far as our understanding of what inflammation and phagocyte recruitment actually means. C3B acts as an opsonin, and again, opsonins tag pathogens for phagocytosis. This is again not a concept that is new to us, something we've discussed in the past. What I do want to touch on now is the formation of the membrane attack complex, or the MAC. So the MAC is a complex of proteins that form a pore or a hole in the microbe cell wall. It is activated by complement system components, and these activated components, again, allow for the holes to be formed in the cell wall. Once these holes have been formed in the cell wall, that allows for water and salts to move in very easily into the cell through these openings that have now been generated. Because of that, the pathogen eventually swells, becomes full of water and swells, and eventually bursts open. So this leads to the destruction of that pathogen. The membrane attack complex is effective for gram-negative bacteria. There are also examples of parasites that can be targeted with the MAC, as well as viruses that can be targeted with the MAC. Now, as I mentioned, gram-negative bacteria are the prime targets of the membrane attack complex. In particular, gram-negative bacteria have a very different cell wall structure compared to gram-positive bacteria. Gram-positive bacteria have a much thicker peptidoglycan cell wall component, and again, gram-negative bacteria have a much thinner cell wall component with a different composition. And it turns out that this thinner component is much more easily accessed by the membrane attack complex proteins. In this particular slide, what you're looking at on the left-hand side is cells that are alive and functioning. And then on the right, these are cells where you have the membrane attack complex forming those holes and causing the cells to burst, leading to the death of the cell. Okay, so transitioning just a bit with the material that we've discussed so far, we looked at the beginning of the lecture functional protein classes that complement system proteins can fit into. The last category we discussed were regulatory proteins. It's very important to be able to regulate this system so that there are not prolonged periods of inflammation in the host system, which can eventually lead to damage of cells and tissues. So there's a whole category for these proteins that help regulate this process. There are, however, 2 ways that regulation can occur. There are passive mechanisms, and passive mechanisms are where the proteins themselves are just very labile, meaning they tend to be unstable, and undergo spontaneous inactivation if they are not stabilized. So if you have a protein component that itself is very unstable, it can essentially fall apart, degrade, and no longer be functional very easily. This is a passive mechanism of regulation. However, an active mechanism of regulation would be where you had specific proteins that have the ability to turn off complement system function. This particular slide comes from a review article. What I want to highlight for you are the components that are in black, either black ovals or black squares. These are all regulatory components that play a role in regulating, basically being able to turn off the complement system. What I want to highlight is that this regulation can occur at lots of different locations in that complex biochemical cascade. The regulation can occur before the assembly of convertase, that is that C3 convertase. So, for example, if you have the C1 macromolecule dissociate, that means it can no longer interact with an antibody and cannot trigger that activation pathway. So that's a way of regulating the process. You can also have regulation after the assembly of convertase, the C3 convertase. If it occurs after, then you would need to stop something downstream of that. For example, binding to C3b and inhibiting its activity. Lastly, another way to regulate this process is to stop altogether the assembly of the membrane attack complex. And lastly, I want to leave you with a concept that will actually transition us into our next set of lectures coming down the pipeline. It turns out that many pathogens have evolved mechanisms for evading the host immune response. So they have ways of hiding, getting away from, not being able to be recognized by the host immune system. The complement system is one of those facets that many pathogens have evolved ways to evade. So, for example, if you look at this figure, the very first component in the figure is that there are plenty of bacteria that have a peptidoglycan component or a capsule component. And this is something that is mostly a polysaccharide coating and acts as a physical barrier. This physical barrier prevents C3b binding, but also prevents MAC insertion. So the evolution of the peptidoglycan and capsule components basically benefit those particular types of bacteria because the MAC cannot insert itself into those cell wall components, and C3b cannot bind overall. In addition, some bacteria have long polysaccharide chains known as LPS, and these are cell wall components. These too act to prevent MAC insertion. So any bacteria that has evolved this particular component on its exterior surface is going to be able to evade the complement system by not allowing the MAC to insert. And then the last example that I want to give you is the one right in the middle of this slide, where you have this sort of star-like looking structure known as C4BP. Now, it turns out that this particular component is found in association with different types of microbial pathogens, and this component is what's known as a molecular mimic. Now, it is a molecular mimic because its structure overall resembles a regulatory protein. The regulatory protein would essentially turn off the complement system. So some pathogens have a protein, again, that is a molecular mimic and acts just like a regulatory protein would in turning off the complement system. So this is, again, just a little hint of some of the things to come down the pipeline when we begin to discuss putting all of the pieces of the puzzle together, looking at the 4 categories of pathogens, and then talking about how those pathogens can actually evade the host immune response.